Okay, good morning, everybody. Hey, you said good morning back. That's a first for me. In Canada, they don't usually bother to reply when you say good morning, but usually they just look a little grumpier than usual. Okay, so welcome. We have a few hours together, and I'm going to try to explain some basics of disease dynamics, within host disease dynamics. And in the middle of that, there's going to be a pretty big detour where I'm going to talk about branching processes, okay? So I'm going to try to pitch this. It's hard to know where to put it because talking to some of you guys, some of you are biologists and some of you are postdoc or faculty level mathematicians. So it's difficult to know exactly where to pin this. So we'll just have to see how it goes. It's very important that you guys talk. Okay, I noticed the last week when I've been in here, it's been a little quiet sometimes, and I know you have a lot of, this is a lot of lectures, right? Coming all at one time. But it's very important that we have some kind of discussion going here, all right? So I want to encourage you guys to speak up, okay? There will be some problem sets during the classes, so I'm not going to speak for the entire hour and a half now, okay? I'm planning to talk for about half of the time, then there'll be some problem sets, and what I want to encourage you to do is to get together into 2s and 3s. You don't have to stay in this room. Okay, we'll have an agreed time when we come back to work on the problem sets together. And so pick your friends or who you're comfortable working with. If you're a biologist, maybe you need to find a math person to speak to. That might be helpful because I wrote them in a sort of a mathematical way. I wasn't completely sure what the best thing to do was, but then I decided this is a math course, so I'm going to write it in a mathy sort of way, okay? But I'll be around. Samit is around. Pranay said he would help, and Somdata I'm sure will be back in a minute. All right, so let's get going. Okay. Oh, by the way, don't call me sir, okay? Only policemen call me sir, all right? No one else calls me sir. All right. All right, so we're going to talk about within host viral dynamics. Oh, it's fine, it's fine. Within host viral dynamics, and specifically we're going to talk about HIV. So I just wanted to give a short background on HIV. Why did I pick HIV? Well, HIV was the first disease within host viral population dynamics made a big impact on the science of the infection. Okay, so that's why I picked HIV. There's been big developments more recently with Hep C. In fact, arguably with hepatitis C, they actually had a bigger influence on how we treat. The mathematical modeling had a bigger influence on how we treat. But that really came about because of the experience that was gained with HIV. Okay, so I'm going to present an HIV-centric set of things, but let me tell you, just about any virus you want to study, you're probably going to end up with a model which is similar to this, and then you're going to modify it to fit what you have, okay? So HIV is a good place to start. So let me just give a little background on HIV. I haven't updated these slides in a while, so they're slightly out of date. But essentially, this was from 2013, I think. Yeah, 2013, so these are estimates of how many people are living with HIV, and there are large numbers of people worldwide, and they are certainly higher now than they were then. Okay, this is a good thing in a sense that they're higher now, because that means people aren't dying. Okay, so the persistence of large numbers of infected people is actually reflecting that treatment is becoming more available, and importantly, treatment's becoming more available where the bulk of infected cases are in sub-Saharan Africa. Okay. Okay, how many, I don't actually know the answer, how many people are infected with HIV in India? Okay, he's going to Google it. Okay, raise your hand when you have the number, okay? All right. Okay, so HIV has actually been with humans, so okay, so here's a question. Where did HIV come from? Africa, but what in Africa? From monkeys, yeah, from monkeys. SIV is an infection of monkeys, okay? It became HIV when it jumped to humans. The most commonly believed story for jumping to humans is in Africa, people will hunt monkeys to eat periodically. They don't do this all the time. And those people, obviously, butchering the monkeys, came into a large amount of contact with infected monkey blood, infected with SIV, and if one of those people was immunosuppressed, then at that point you could imagine the blood could have come into their system and the virus could potentially have taken off. So usually we're not very susceptible to animal diseases, but there are notable exceptions. Somebody else tell me a disease with an animal reservoir.
Ebola.
Ebola, yeah. But actually, do we know what the reservoir is now? Is it bats?
Gorillas, but
I'm not sure it's gorillas. Okay.
maybe.
Rabies. Rabies is a good one. So rabies is actually endemic in bats. So don't touch bats, right? That's the story. But the rabies is very common in bats, and then dogs and humans can pick it up essentially from bats. But many diseases we're not terrifically susceptible to. Diseases of farm animals, for example, you don't really usually pick up. Anyway, so phylogenetics, which is, basically looking back down the family tree using genetic information from now, kind of points to the first transfer, or the current transfer at any rate, from monkeys to humans around the 1900s, okay? So around 100 years ago, give or take, okay? There is some debate, and obviously this is a little imprecise. But looking in the freezer, looking at tissue and blood that was stored in freezers in Africa, it was discovered that there were actually cases of HIV back to 1959. So some doctor had an unusual patient died, they stored some tissue, and then it was found and thawed out 15, 20 years ago, and we're back to 1959 for the first sort of confirmed case in humans. When AIDS became a thing, when it came to the United States, roughly speaking. So there were a number of individuals who were men who MSM is men who have sex with men, or we can say gay men, and also hemophiliacs who receive regular blood transfusions. The blood was not being tested for HIV because no one knew it existed, and this was picked up in the United States in sort of around 1980 or thereabouts. And I actually remember as a kid, so I was 6 in 1980, I remember watching a science show. There was a science show that would come on, and I had to ask my parents to stay up until 8 o'clock to watch the science show. And they had one on this disease, which was called AIDS. And I think I watched that in around 1982. It was all very scary, because nobody knew what it was, nobody knew how it was transmitted. All they knew was a lot of people were dying from this disease. Okay, the virus was isolated and named in 1983 by a group in France and a group in the US, more or less at the same time. There was a great deal of fear about the disease at that time. I also remember this when I was slightly older, getting official information from the government on how to avoid HIV. Then the treatment didn't come until a little bit after that. And the early days of treatment was essentially, it would keep you alive for maybe 6 more months to a year. Okay, and then the treatments gradually improved until, do you guys know who Magic Johnson is? Magic Johnson? Yes, you know it is, but Magic Johnson is a former NBA basketball star, and he's been HIV, he announced he was HIV positive in the early 90s, and he's still around and he's fine. So as long as you take your drugs, you're not going to die of HIV, seems to be the story. The drugs have some side effects. It's not a good thing to be infected with HIV, but that's the story. Anybody have any questions about that? No, okay. All right, you really don't have any questions. There must be one question somewhere. No? Yes, good, thank you.
What was it named before it was officially named?
Yeah. Okay, I can't remember. It did have a name, it was CT something, and there was a French name that kicked around as well. When they were first naming it, there was some uncertainty as to whether the French team and the US team had actually gone there, had identified the same virus. But the name that stuck in the end was HIV. Yeah. Okay, how many HIV infected in India?
In 2010, there were 2 million, 2.2 million.
0.2 million. Just 200,000? That's the official number. Yeah.
2008 and 10.
Oh, okay. Maybe a little higher than that, I would think, but yeah. Yes, sir.
How many different strains are recognized currently?
There are very many strains. There's the basic distinction between type 1 and type 2. Sorry. I'm not an expert on this, so this is all coming out of the back of my mind, but there's essentially a kind of HIV which is mostly just present in Africa. We don't know that much about it. It's only in West Africa. Yes, Sundata.
So there are only basic 2 types, HIV1 and HIV2.
Yep.
HIV2, very few people get it very
Yeah. Yeah, HIV2 we don't know much about, but I think it came from a different animal. I don't remember the names of the monkeys, There are a lot of monkey names, but there was HIV2, I believe came from a different animal than HIV1. And it may be a completely independent epidemic. It's primarily in West Africa. HIV1 is what we have that spread through the rest of the world. It's relatively young disease, but it's highly mutable. So if you plot the edges, even within one patient, you can find a phylogenetic tree of some appreciable size, and then the global distribution is big. Yeah. Thanks, Madhava. Yeah, I forgot you were an expert. Yes. Yeah, it's a virus. Yeah, it's a virus. So viruses need the host cell to replicate. So HIV can only replicate itself when it's inside the host cell.
And there are only few means where it can be communicated.
Yeah, sex and blood.
Is there any specific reason for that?
It's very weak. So it doesn't primarily infect the lungs, so it would be difficult to cough it. I think salivary transmission is sort of one of those things that's theoretically possible, but in practice never happened, right? Yeah. We're adults, we can talk about this, right? So insertive oral sex is extremely low risk. Okay, extremely low risk. Okay. And I think it's essentially not measurable. If you look in a paper, they'll say something about this, but it's essentially receptive oral sex is low risk, but still very, very rare, but you can transmit that way. Yeah. Yeah.
It doesn't spread through saliva, then how come would be spread through orally?
Yeah, how would it do that? So you could have a cut in your mouth. That's what I would go for. A cut or a sore or something like this. Yeah. Okay. All right. Okay, so the virus predominantly infects CD4 positive immune cells. So these are T cells of the immune system. So we don't actually need to, I'm not going to go into any immunology, really. But CD4 T cells are the helper T cells and macrophages of the immune system. Primarily, it infects T cells. So T cells, because they play an important role of your immune system, then eventually, as you lose the T cells through the infection, eventually this is what causes you to become sick. So HIV itself does not kill you. It's infections which you develop because you have an inactive immune system. Okay? So, well, we'll see what happens to the T cell counts over time, but basically they're dropping. Okay? When you get infected, you should find the virus in the blood within about 10 days. Okay, so free virus will be in the blood in around about 10 days. It might be somewhat longer for some people. Yes?
You can't actually test for HIV until after chemotherapy, right?
No, that's not true anymore. In the old days, you had to. This Australian guy I knew once was at risk for HIV, let's say, and he was worried because he had gonorrhea, but he had to wait 6 months back in the 90s to have an HIV test. Yeah, but these days, so a blood RNA test is almost, will be the tests are better, so that would be good. The old antibody tests, which are still the gold standard diagnostic tests, you're right, they take longer. But if you go to your doctor in Vancouver and say, I'm worried I may be at risk for HIV, they'll immediately give you a test just to see if you were infected from before. But then they'll schedule follow-up tests over the next couple of months. Blood tests, yeah.
Isn't there a drug that you can take right after you think you're going to...
Yeah, we'll come to that, yeah, that's a good point, yeah. Okay, once you've been infected for 10 days, or at least if you're a monkey who's been infected experimentally for 10 days, the virus is everywhere in your body, okay? So this is one of the reasons it's difficult to cure, is because you find the virus in almost every tissue. Certainly, after a year of infection in a human or a little longer, when they've been able to look, they've found virus in almost every organ, one way or another. Your immune system permeates your body, so it's not surprising that you're finding the virus throughout the body. Okay. All right. Oh yeah, this is a picture of a schematic of an HIV virus, and yeah, you can't read that. I think I have a better picture coming up. Okay, I'll try to point out some of the Well, actually, maybe I can point it out. Okay, it's an RNA virus, so this is RNA. So how does RNA, so when it infects the cell and the RNA comes out, the first thing that happens is it has to get the RNA transcribed back into DNA. Okay, this is just how it works. It doesn't go directly from the RNA in here to the protein. Okay, so the RNA here has to be transcribed back into the DNA by an enzyme called reverse transcriptase. So the RNA here is packaged with the reverse transcriptase in the viral particle. Okay, so the reverse transcriptase will translate the RNA back into DNA, and then the host cell machinery goes DNA, RNA, protein as usual. Okay? If we look on the surface of the cell here, these molecules which are indicated as being very regular, by the way, they're not, just all over the place, but these molecules are called GP120, and GP120 is what the virus uses to bind onto CD4. CD4 is on the outside of the T cells and macrophages, and then once it's bound, it's able to find a way in from there through processes that we do not completely understand. Okay. Oh, I don't know why that was there. Okay, yeah, oh, this is a much better picture, yeah. So here's the virus with the GP120 on the outside. It comes in and it binds to CD4, and it also requires a co-receptor molecule, but let's not worry about that. Okay, here is the RNA. The virus kind of fuses itself inside from here to here. This is the part that we don't completely understand how it does it, but it does it somehow. And now here is the free RNA. Okay, the reverse transcriptase enzyme works on the free RNA, reverse transcribes it into DNA. Yeah, the DNA is packed up in the nucleus. Yeah, so it's just inside the regular DNA of the cell. It gets transcribed now back into mRNA and then into proteins, and somehow the proteins self-assemble to form the virus. Okay, and the virus buds off. Okay, and then this guy can go and look for another cell to infect. Okay, so without a cell to infect, HIV is nothing, right? It needs, and any virus is nothing. It needs a cell to have this process. This is all just background. I'm not sure you really need to know any of this, but I always feel like you need to know 10% more than what you really need, you know, so that you have some, if somebody says something, you kind of understand it. Okay, so getting on to measurements, okay, so the kind of clinical measurements people can make. This is a horrible graph, okay, but, so let's start off by looking at the red curve. So the red curve is the viral load. So here is the viral load. This is the number of copies of the HIV RNA, all right, which are found in a milliliter of plasma. So you take the plasma from the patient, and you have a test which counts how many copies there are. And this is a trick scale down here. Okay, so we have the early part of the infection, and then we have like a long-term progression. This is untreated infection. Okay, so in the early days of infection, you get a jump to a peak viral load in most patients, okay, of around 10 to the, I know you can't see that, so that's 10 to the 6 viral copies per milliliter, okay, RNA copies per milliliter. Okay, but it then drops, okay, and then it will reach kind of a more or less stable state. We call this the set point. Okay, so the viral set point, and that set point will actually stay around the set point for quite a long time, for several years, until after sufficiently long, there is sufficient destruction of the immune system that opportunistic infections come in, and this is AIDS over here, so this is when the patient will die. So now let's look at the CD4 lymphocyte. Lymphocyte means essentially immune cell, okay? Let's look at the CD4 lymphocyte count in cells per millimeter cubed, okay? Okay, so most of us in this room will have, we're uninfected, right? We'll have somewhere around, say, 1,000 CD4 cells per milliliter, okay? Millimeter cubed, and so some of us won't. There's actually quite a lot of natural variation in that. So some people are more, just uninfected people will have 600, some people will have 1,500, okay? But there's quite a bit of variability. But let's say they start off around 1,000, and then it drops. Okay, so as the viral load comes up, the cell count drops. And this is presumably because the virus is infecting the cells, making more copies of itself. This makes the cells really sick, and the cells die. Okay, so we get lots of virus production, lots of cell death. Okay? There'll be kind of a low point around here, and then it picks up again. Okay? And then when the viral load is more or less in this kind of steady state, okay, the T cell count, this figure shows it as sort of dropping in a log linear kind of fashion. Honestly, anything, the T cell counts that people measured on patients who had AIDS in the days before we had any treatment, the T cell counts would be all over the place. The whole system completely fails at this end. Okay. There is no good model of AIDS. Nobody models that. And actually, it's pointless to model it anymore, because if you have a patient with HIV, if they had AIDS, you would treat them, you would knock out the HIV. We have good treatments. You would knock out the HIV and they would stabilize most of the time. The number of patients dying of AIDS is now quite low, okay, as long as the therapy is available. All right. Yeah, so therapy. ART is anti-retroviral therapy. We've had good ART since the mid to late 1990s. These days, if you are an HIV patient and you take your therapy every, I think it's actually more than once a day, three times a day you take your therapy, your viral load in your blood will go down so low that it cannot be detected. Okay? So the viral load will be so low. However, it's not cured. If you stop taking your medicine, the HIV will come back. Okay? There's been no exceptions to that so far. And your immune system will rebound. So your CD4 count will climb back up, and you'll essentially be healthy. So the only thing as long as you take the medicine every day, the only thing you really have to worry about is the side effects of the medicine, okay? Which are there. They get better because they're developing better drugs with less side effects. Depending on where you live, you may have to worry about the cost of the treatment as well. Okay, the treatment is still relatively expensive for resource-limited countries. Yes? Yes.
Can you still infect the people?
Yeah, so this is actually very controversial. So the question was, can you still infect other people if you're on stable therapy? So in Switzerland, about 10 years ago, the Swiss public health authorities made a statement that if your viral load was suppressed and had been suppressed for a year or some period of time, maybe 2 years, then you didn't need to worry about being careful with sex anymore, okay? So they said, and this was actually a very controversial statement at the time, but they said it, and I don't know what happened afterwards, but, but the general view is if your viral load is suppressed, it's quite unlikely that you'll infect anybody else. However, I personally feel like you should still tell the person you're planning to have sex with so they can make the decision rather than you making the decision for them, but that's a whole nother story, right? Yeah. Anyway. Yeah. Oh, yeah. How many people have been cured of HIV? No, you're not allowed to Google for the answer. You have to tell me.
A few, a handful, right?
Should be none.
A handful, none. Yeah, answer is actually one. One patient. I'll just quickly tell you the story. He's called the Berlin patient, although his name is now public. But initially he was private, and he's called the Berlin patient. So this guy, you could say he was lucky, or you could say he was unlucky. So he's an American guy. He was living in Germany, and if I get the story slightly wrong, then just correct me later. But he was living in Germany, and he was HIV positive. He knew he was HIV positive. He was taking drugs. He was okay. And then he got leukemia, I believe. Okay. The therapy for leukemia is a bone marrow transplant. Okay. But before they give you the new immune cells, right, they have to kill off all of the immune cells you have. So they killed off all of his immune cells by radiation. Okay. And then the bone marrow transplant they gave him was from a donor who was HIV negative, obviously, but also from a donor who there are people primarily living in Europe who have some small natural resistance to HIV. I'll get to that in a second. They have small natural resistance to HIV, and you can find these people. They gave him a transplant from a donor in that group, okay? And after however long, his HIV went away. So you could say he was lucky. I don't know. If you have HIV and leukemia, I would say personally you're unlucky, but he doesn't have HIV anymore, and I believe he doesn't have leukemia anymore. So yeah, it worked out. There have been other patients where similar things have been tried, patients who had HIV and then subsequently needed a bone marrow transplant. And as I understand it, none of those people have been cured. So it seemed to be a lucky break for this guy in a way. Okay, so essentially one patient has been cured of HIV. This business, by the way, of the resistance to HIV infection, this is also kind of an interesting little piece of science. So when I showed you the virus docking with CD4, okay, so it binds to the CD4, and then there's a co-receptor. Okay, some people have a mutation in the co-receptor here, okay, and it's common. I think around 10% of people in Northern Europe are heterozygous for that mutation, so they have one normal copy and one mutated copy. But obviously, if 10% are heterozygous, around 1% are homozygous for that mutation, so they have two mutated copies, and those people seem to do slightly better in terms of their progression to AIDS is much longer, and they seem to have a lower viral load and things like this. Okay, and the reason that mutation is in Europe and not elsewhere is believed, at least the last thing I read about it, is believed to be because of the plague. So we had, in Europe, there were these terrible plague epidemics that swept across Europe, and it also appears, it's believed that it may confer some small resistance to the plague, and so it was selected for in Europe. Just an interesting piece of science if you like that kind of thing. Anyway, okay, so this is a plot of what people die of in the US. So this is men between 25 and 44 years old, okay, and what are their causes of death? Okay, and there was lots of curves on here for different ways that you can die. But you can see that HIV infection between about 1991 and 1996 was the leading cause of death for young men in the United States. Okay, and that precipitous decline starting around 94, 95 is because of treatment. Okay, so because of treatment, they're doing much better, and basically, it's dropping off. Okay, just a comment to the young men in the room. If you're over 44, you don't have to worry about this, okay? But unintentional injury is really high, so take it easy crossing the street, okay? Anyway, all right. So this big drop-off was due to was due to the introduction of the medicine. It's very, very good.
So I suppose that, I mean, there must be other causes of death, of course, but it looks like it actually constitutes a significant drop-off in death.
Yeah, I think it would have been noticeably.
The death rate is low.
Yeah.
But very measurable difference.
Yeah, and without the therapy, what would have happened? Without the drugs, you can see the climb there, right? That was a rapidly increasing cause of death. Now, these are all causes of death. I don't. There are a few missing, but they're all down here. Right. I don't think they add up to all that much. If you're a young man, you're probably not going to die for a while, you know. Yes. Homicide. Yeah, here it is. Oh, murder. Murder most foul. That's when somebody kills you.
Yeah, but how is that related to HIV?
It's not. These are all causes of death. These are all causes of death. HIV is only one choice. You can be killed in many ways. Yeah, yeah. Liver disease. You know, I mean that's probably related to hepatitis. Yeah. Yeah. Okay. All right. Oh, diabetes. Yeah, diabetes. Yeah, he came back. Okay. Diabetes is here. The purple line here, right? Yeah. Okay. All right. The drug actions. Just briefly, there are different classes of drugs. Protease inhibitors, sorry, I'll go through them in order. Integrase inhibitors prevent the viral, sorry, I've got them in the wrong order again. Okay, here in order. Entry inhibitors prevent the virus from getting into the cell. Okay. Reverse transcriptase inhibitors prevent the virus from reverse transcribing the RNA into DNA. Integrase inhibitors prevent the proviral DNA, the viral DNA, from getting into the nucleus and being integrated, okay? And protease inhibitors prevent the mature viruses from escaping from the cell, okay? So those are the four main classes of drugs that we have. The first two to come along were the reverse transcriptase inhibitors and the protease inhibitors. And then after that, the entry and integrase inhibitors were coming a little bit after that time. But these days, most people will take several different drugs. Their therapy will be 2 or 3 drugs, probably 3, from different classes. Okay, so in the event that the virus was to develop resistance against one, it probably wouldn't be able to develop resistance against the other. Okay. All right. Treatment is very, very good. Now, so this guy actually lives in Vancouver. He's the big man of HIV epidemiology in Vancouver. And I'll just show you this graph here. So this is the decline in Vancouver, British Columbia, in Canada, where I live, in the number of deaths per 100,000 people. Okay, this is the decline which was caused by the drug. So this is HIV deaths. Sorry, this is HIV diagnoses. So what was happening is, as people were becoming virally suppressed, this was your question, Rimjim, right? As people were becoming virally suppressed, they became less transmissive to other people, okay? Now, you could argue, actually, this is because everybody was being very safe with sex, right? So everyone suddenly became very careful. But look, here's syphilis. So if people were being very careful with sex, syphilis would not be going up at the same time as HIV was coming down. So this is treatment reducing transmissibility, okay? So there's a lot of benefits to treatment. You should treat people because it makes them better. And actually, if you treat people early in the infection, they have better outcomes, okay? They're healthier. But also, it has a public health benefit in that treatment reduces transmission to new patients. Yes? Sorry, I couldn't quite hear that.
You can actually edit the genome which goes for the particular receptor and using CRISPR-Cas9.
Oh, do gene therapy?
Yeah.
Yeah, I don't think anyone's tried that, but it would be interesting. I don't know. It seems like that would be a pretty big tool to apply to groups of people, but yeah. We talk about it at tea, okay? Yeah. All right. All right. The other thing Jim meant, or somebody asked the question, post-exposure and pre-exposure prophylaxis. There have been trials of this. So the first thing is pre-exposure prophylaxis. Basically, you find people who are likely to become HIV infected. Okay? So men who have sex with men, female sex workers, so prostitute, we call them sex workers, and maybe a few other groups. And you ask them to take the medicine every day. It's a relatively low dose, but it's been shown in trials that this is effective at reducing transmission. Okay, so the drugs also prevent initial infection if they're taken regularly. That's pre-exposure prophylaxis. Post-exposure prophylaxis has been around for a while. If you're a nurse or a doctor and you accidentally come into contact with a patient's blood, then you can go to the pharmacy in the hospital and request 1 month of HIV treatment, and they will automatically give you this. And you take this even though you don't know if you're infected. You don't know what happened, but you don't want to be infected, so you take this. And this is now being prescribed by doctors just for members of the public who believe they may have been exposed to HIV. Okay? So the drugs are in use as prophylactically. Okay. All right. Okay, so just to summarize, HIV is a virus, and it infects and depletes CD4 T cells. It takes around 10 years to develop to AIDS, where most people will die. One person has been cured, which is the same record that the human race has as for rabies. People who get shots for rabies, we don't know if they're infected or not, right? Continuing transmission worldwide, lots of treatments available. The treatments are really good. Development of a vaccine has been slow. A lot of people thought there would be a vaccine in the 1990s. There's still no good vaccine right now. There are candidate vaccines and ideas, but there's no real good vaccine. Okay, that's the end of the bio part. And actually, there's not much more biology to come. So I hope you enjoyed it. Yeah, I highly encourage you to read more. If you can go to the Wikipedia page for HIV and delve into that, there's an enormous amount of information available. Also, there are some HIV prevention organizations that actually, their information that they give to people is very, very good in my opinion. Yeah. Okay. All right. So here come the equations. All right. So this is the standard model of HIV infection, and some people even call this the standard model of virus dynamics. Okay. So it's a deterministic differential equation-based model. And the way I have it written right now, there's no treatment, okay? All right, so here we go. So we start off with a population of target cells. So T will be the population density or number of target cells. So T is equal to the target cells, okay? And the patient produces those at some rate lambda, okay? So in the particular case of T cells, target cells for virus, in the particular case of T cells, your thymus, which is up here somewhere, is producing T cells all the time that come out and replenish the population. So we have a rate lambda, okay? So this is new cells produced, okay? And these guys also have a natural death rate, okay? So this is a death rate. So you can see that in the absence of the virus, we'll have a population lambda divided by D of the target cells, okay? Now what can happen to target cells is they can become infected. And they come infected when they come into contact with a virus. So you have to have a virus and a cell. This is like a chemical reaction, right? So you have a virus and a cell come together, and that will produce an infected cell. And this K here is essentially like a chemical reaction rate for this reaction, okay? So when the infection happens, we produce what I've always called a T star. So T star is an infected cell. Some people use I for infected cell. The T star is the infected cell. Okay. Okay, and so they're being produced at rate kVt. Okay, and then these cells die too, and they die at a different rate because they're infected, they're sick. Okay, so this is a mass action infectivity. Sorry, getting excited here. Okay, so we've got mass action infectivity produces new infected cells, which then die at some rate mu. Okay, yeah.
New cells produce is not a function of birth rate and T.
Okay, this is a big simplification. All right, it's a big simplification. But broadly speaking, lambda is the thymic output, so it's how many new T cells are being produced per day. Okay, and D, 1 over D is their average lifetime. Okay?
But there's no ramping up of production when you have a low count.
No, that doesn't seem, there is, but it's not very important. Okay.
I mean to say, lambda is not written like lambda is equal to nu T, where nu is the birth.
No, it's not. No, this is not a birth rate. This is not the T cells making more T cells. There are stem cells in your bone marrow that are just going, okay? That's how you should think of this, okay? Now, when the T cell count drops a lot, yeah, there's experimental evidence to show that this is a bad model. But this is, remember, what's a mathematical model supposed to be? We abstract and simplify as much as we can, right? So this is the standard model. There are certainly papers out there where they try to really investigate very carefully these terms here, okay, and try to replace them with something better. But this is the simplified standard version, okay? All right. Okay. And what do infected cells do? They make viruses. Okay? So V equals viruses. So dV dt, the rate of change of the number of viruses, we have a production rate P times the number of infected cells. So each infected cell is producing P viruses per day. Okay? So this is the viral production rate. Okay, and then the viruses are cleared at some rate C. So C is the viral clearance rate. Okay. Yes?
What is the clearance rate?
For the viruses? Because if you take a virus and put it in your blood, it will actually get filtered out very rapidly. And they do great experiments with mice and monkeys, and they can measure this beautifully. What I'm going to show is how this parameter was estimated for humans. Okay, so I'm just going to quickly show that. Okay, but that's actually one of the most important terms, as we'll see. Yeah. Any questions about the equations? Sorry? Yeah, then they've been destroyed. That's it. They're done. Yeah, they're done. Any questions about the equations? There is one question that often comes up. Okay, I'll ask the question. All right. Okay. When a virus infects a target cell, yeah, I made an infected target cell, and I subtracted that from the target cells, right? Should I have subtracted it from the virus as well? Yeah, we lost a virus there, right? So now, okay, all right. I should write that, yeah? Do you agree I should write that? Because we're losing a virus at the same time. Now, no one, okay, this is not true. Most people don't write this term here for the following reason. You have small numbers of target cells and giant numbers of viruses. So losing one virus here is just not a relevant thing to include in the model. And it makes calculating the eigenvalues and so forth a bit of a nuisance. Like you have to solve more complex equations. So generally speaking, my recommendation is, unless you have a good reason to leave that there, don't do it. Okay, just let's not write that term. Okay, I've said to students before, the cool people do not write that term. Okay, there are a few people that like to keep it there because it makes them feel better, but it's crazy if you ask me. Okay, all right. Okay, so we got this model, and here's the measurements. Here's a couple of patients that have been fit. This is time in days from 0 up to 80 days, okay? And this is initial infection. So we have here measurements of viral loads, and this is what this model is doing, okay? This model is passing quite nicely through the viral load measurements. Just a comment here. It's quite possible with this model to fit individual patients. However, when you fit the parameters vary a lot between patients. There's a lot of interpatient variability according to this model, and that's never been successfully really dealt with in my opinion. But people are actually quite different, and people's responses to the virus are quite different. So it's quite normal to find a pretty big range in these parameters, okay, when you fit the patients. So it's very difficult to fit all the patients together, but to fit them individually works fine.
Can some of these parameters be measured independently of the fitting process, things like lambda or d?
In principle, yeah. In principle, yeah. And for monkeys, so for experimental infections, people have tried to do some of that stuff. Yeah. Okay. All right. So just quickly, linear analysis. So Bard made you do this, right? Yeah, and then some data made you do this. So this model, as I have it written down, supports two steady states. Okay? There is an uninfected steady state, where you can see there's no infected cells and no virus. And there's an infected steady state where we have more complicated expressions for amounts of T cells, infected cells, and viruses. Okay? So there are two steady states. So what do we do with steady states? We linearize around them and we look at the local stability. Yeah? Okay, so looking at the uninfected steady state in the top left, okay? All right, so now we perturb around the uninfected steady state, so we introduce perturbation variables that have T hats, right? If you want to, you can just jump straight to the Jacobian, I don't care. I'm not gonna get upset. Okay, so this is the Jacobian matrix at that point. Okay, so my perturbation variables look like this. I calculate the eigenvalues of the Jacobian matrix, and I get, essentially, I get this should be 2 and 3. I get 3 eigenvalues. One of them is just minus D, okay? The other ones look complicated, but if I analyze this quadratic solution here, then it's quite simple to find a stability condition. So by stability, I mean that we have negative eigenvalues. So the condition that the eigenvalues are negative is this, okay? So this combination of parameters has to be less than 1, okay? If I have this, then I have that the uninfected steady state is stable. So what would that mean? If the uninfected steady state is stable, what does that mean, practically? Virus what? The virus will die out. You go back to the uninfected steady state. It's locally stable. You introduce a small perturbation of virus, and you get sucked back to the uninfected steady state. The virus dies out. Great. Yeah. Okay. Now looking at the... Hello, Pranay.
Is that, for real parameters, is that locality?
It actually does, yes. Yeah. And this, I'll mention it a little bit more in a second, okay? Yeah. Okay. Just looking at the infected steady state, okay? So now again, I make perturbation variables. I do a little algebra. I get my Jacobian matrix around the infected steady state, okay? I calculate eigenvalues. They're awful, okay? But what I can do is I find the stability condition is the same. So the it's the converse of the previous one. The infected steady state is stable exactly when the uninfected steady state is unstable. So basically, there's only two things that can happen. Either this quantity is less than 1, and then you never get an infection, or this quantity is greater than 1, and then if you induce a little bit of virus, okay, I'm not proving that just exactly on this slide, because this is a global statement, but it's still true. If you introduce a little bit of virus, you end up at the infected steady state. Okay, so there's only two things that can happen, and we have exchange of stability between them. It's very simple, right? It's very simple. Okay, so we can call this quantity an R0, a basic reproductive number, okay, for the virus. Do you know this concept of R0? Yeah. Okay, and you can also show that this controls global stability. The Lyapunov function is not difficult to construct. In fact, I almost put it on the problem sheet. Okay, all right. Okay, and so if I plot some solutions, I just made up some parameters that I thought were sort of roughly correct, and I can plot, you can see in the top left, the blue line is the T cell count, or uninfected target cells, and they're dropping and coming back during the first few days of infection, and the viral load is on a log scale, because the viral load's very big, and it's climbing up to quite a high peak and then dropping down a little bit to some steady state, okay? All right, and if I plot in the phase plane, t against the log of v, I actually see that it's a spiral point. It's quite nice, right? So I have complex eigenvalues with a negative real part, negative real part locally to here, so I get a spiral point. And that spiral point corresponds to the oscillations that you see in the plots of v. Okay, all right, so this is called the basic model, or the standard model of virus dynamics. And this is the model that if somebody came to me and said they wanted a model of Ebola infection, guess what I would do first? The first thing I would do is I would just get this model out and start trying to fit parameters. Fortunately, nobody asked me to do that, but could happen, right? Okay, so I just want to talk a little bit about the history of how this model really was helpful in the 1990s. So this is 1995. So this is the era of the beginning of therapy. So they have therapies now that they can give to patients, okay? And what we're seeing here is 3 patients, and on day 0, they're given drug, okay? And what you can see is the viral load starting off kind of level in their blood is dropping during, after the drug is taken, okay? It doesn't go to 0. Okay, it doesn't even go undetectable. You can see it's still detectable here. Yeah, okay. But this patient, kind of the same story, and this patient, similar kind of story. Okay. So that was the data. Okay. So the model, which was at that point not considered the standard model of virus dynamics, it was just some model that someone came up with. Okay. The model was then applied to this data. Okay. So if you assume that you're at the infected steady state before you're here, okay, you don't really need that assumption, but if you assume that you're there, okay, and then you assume that the virus is turning over much faster than the infected cells, okay, so this is actually an assumption. You assume there's lots of virus and not very many infected cells. Then what you can do is replace the V equation here with a much simpler equation. So dV dt is now equal to P times the steady state of T stars. Okay, so we replace the T star with its steady state value, because we say it's going to move slowly when you start treating, and the virus is going to move fast. Okay, but the clearance rate is still here and the virus is here. So this is a linear equation, right? Yeah, this is a linear equation. This is a constant. This is a constant. My variable I'm trying to solve for is V. So I have dV dt equals constant minus CV. This is like a capacitor equation. Something like that, okay? We start off at the steady state, and we can solve that immediately, okay? We can solve that immediately. This is an exercise you can give to a first-year student, yeah? Okay? Okay, if the drug is perfect, then what happens to the production rate? If the drug is perfect, then the cell can no longer produce virus. So P equals 0, and we just get exponential decay. So you switch on the drug, and the virus immediately suffers exponential decay. Okay, log, linear axes, this is an exponential decay. You read off the slope of this line that you fit through, the first 3 or 4 points here. You read off the slope of that line, and that tells you what C is. Okay, the slope is C, the clearance rate of the virus. If you have an imperfect drug, then your clearance rate is going to be a little bit faster than the slope you observe. Okay, so if you have a drug where this term is still hanging around, and this is the data, you think about where the slope is. Yeah, if the drug was perfect, then it's going to be going faster. So that means that C is going to be bigger than the slope that you observe. Okay, and looking at the slope of this line here, the slope is minus 0.2, and if you convert that into a half-life, that's a half-life of 3 days. Okay, so it's telling us that there's a lot of turnover going on. Okay, there's a lot of turnover. All right, lots of production and clearance. Because there's lots of viruses being produced, that means there's a lot of mutants, which means you have the opportunity for immune escape or drug resistance to come up. And this was actually a big change. In 1995, the predominant view of was that the reason there was so much virus in HIV-infected patients was because it produced a whole lot and then it hung around for a while. The virus wasn't going anywhere. Okay, but this tells you, this experiment, plus just a little bit of modeling or a little bit of careful thought encoded as a model, tells you that the virus must be turning over every day. So actually you're producing a lot of virus and you're clearing a lot of virus every single day, because as soon as you switch off the production, you get a big drop. Yeah? So think about it like a reservoir. You got a lake, yeah? Okay, imagine the lake is really full. So the prevailing wisdom in 1994 was there's lots of virus, the lake is very full because it produced a lot, fills up the lake, and very little is draining out. There's very little clearance, yeah? What this is saying is that's not true. The lake is full, but actually there's a lot of water coming in and there's a lot of water going out as well. Okay, so it's a very different view of the virus than what was around before. Okay, and in a follow-up paper, more or less the same authors as before, they did some similar things where they have a drug now which makes all the viruses still get produced, but now they're not infectious anymore. Okay, so you have non-infectious viruses, okay, but you still detect the viruses in the assay, okay? And looking now at the viral loads in these plots for different patients, and also an experiment where they took the blood and they tried to infect cells in a dish and they saw how infectious it was. You can see the infectivity drops off very rapidly here, okay? So what they can do now is fit a slightly more complex version of the standard model, okay? So what they do is they assume that before treatment everything is at steady state, but post-treatment the T star cells are now allowed to turn over. Okay, so they now allowed the T star cells to turn over. So these T star is still dynamic in this version, okay? But the T cells, the target cells, are fixed. So they don't move on the time scale of the experiment. The T cells are fixed, and we just look at what happens to the T stars and the Vs. So you let the thing go according to these equations with T fixed at its steady state value. Are these linear or nonlinear? This is linear, right? There's no nonlinear term anymore. So because it's a linear equation, you can solve it, and you get a solution which looks something like that. Okay, and you can now fit the viral load data to this. Okay, and you know, with a little bit of careful adjustment of parameters where you think you know them and things that you allow to fit freely. Okay, they obtained that the half-life of the virus was less than 6 hours. Okay, so this is really emphasizing the point that the virus turns over rapidly. There's massive viral production, there's massive viral clearance. There are many, many infected cells being infected and dying every day. And that's what wears out your thymus. Ultimately, that's why you don't have as many T cells after a few years, because it's producing so many T cells and eventually it gets worn out. Now that's getting to your point of why lambda is fixed. Yeah, but here, we're only looking at the first 30 days of infection, so it's probably okay. Yeah. Okay, So I'll just read you a quote from one of these papers. Okay? Sorry, I'll start with the bullet points. Massive ongoing replication and clearance of HIV. It also showed that any drug that you give should work within a few hours. Because it's such a dynamic disease, anything you do, you should see a result within 1 day, at the longest. Yeah, so if you give a drug and there's no effect after a day, well, chances are that drug isn't going to do anything. Yeah. HIV generates, and because there's so much viral production, that means the virus can generate mutants very, very rapidly. So you can generate mutants extremely quickly and therefore escape from the immune system and escape from drugs that we might give. Okay? All right, the failure of the current generation of antiviral agents when used as monotherapy, so just one drug, okay, is the inevitable consequence of the dynamics of HIV replication. So what they're saying there is they were giving patients these drugs and they were seeing the viral load drop, and then after about a month the viral load would come back because the viral replication was not being sufficiently suppressed to prevent the generation of mutants which were drug resistant. So the viral load would go that down, stay down for a month, still detectable, still a lot of virus being produced, and then an escape mutant would be generated and the viral load would go right back up again. Okay? They're saying this is the consequence of the dynamics, the fact that it's a dynamic disease. Effective treatment must instead force the virus to mutate simultaneously at multiple positions by means of a combination of anti-retroviral agents. So you make it so that a point mutation here is not enough. You need a point mutation here, and a point mutation here, and a point mutation here. And then combinatorially, that gets very unlikely. And so this was the breakthrough that came in 1996, and led to that graph that I showed where the number of deaths dropped rapidly, yeah, and then continued to drop because they were treating multiple drugs all at the same time. Okay? Instead of treating with a drug until it fails, and then treating with another drug until it failed, and so on. You treat with all at the same time to make it very difficult for an escape mutant to come up. Okay. All right, so just to summarize, the standard model has been used a lot. It's a big success of math biology. Like if you ask people what's math biology good for, or if someone asks you this in the street, you know, you're just standing there and someone comes up and says, what's math biology good for? Okay, you might pick this as an example. There are other ones too. This is a good one. And people have extended the model in all different myriad kinds of ways, okay, over time. The main criticism of this model is, why does the viral load go up and down? It's because the target cells go down, and that means there's no cells left for the virus to infect. Yeah? And then the way that the steady state is maintained towards the end is a balance between the production of target cells and then being infected and dying. There's no immunity in this model. And that's actually a criticism. So some people call this mathematical immunology. I don't think that's a very good name because there's no immunology here at all. This is just virus dynamics. This is population dynamics of viruses. Okay? So there's a serious criticism of this model is there's no immune system. But with that said, we've had this model over 20 years and it's still sort of doing okay, even without an immune system. You just have to make sure you don't ask it a question about the immune system. Great. Okay. So do we have, are there any questions there? Yes, sir. Thymus transplant. I don't believe that's ever been done. Yeah. Yeah, good question. I mean, then you're just giving more food for the virus to eat.
No, I mean, you remove all the...
How would you do that? Yeah, the problem is the infected T cells are everywhere. Your brain, every organ has HIV in. So this patient in Berlin, I mean, they irradiate the guy, you know. That's very serious. I mean, they're not going to do this for patients, yeah. So simply like filtering the blood or something, that's just not going to do it because most of the virus is elsewhere. Yeah, the blood is just a readout of the virus which is elsewhere in the body, yeah. But I like your optimism. Yeah. Okay, so I made some problems for you guys. So I wanted to distribute the problems. I'd like you to work in groups, okay? This will force you to think and talk together. Some of the problems are a little technical. When Sunny comes back and we can come around and help you, but I have a copy for each person. So we'll take